Hey guys, my name is Anthony, and this is my honors project. So I decided to do a research project, and it's basically exploring a technology called Refractive Index Cytometry. It was a collaborative effort by with the Polytechnique Montreal and the Maisonneuve-Rosemont Hospital, so thank you to them. Now, what is this technology that I'm talking about? Well, it's also known as simply RIC, and it's a vanguard technology, so it's still under development. It's a tool that analyzes cells, and it's basically designed to detect them and to identify them. So this is what the overall RIC setup looks like, but I'll go over its main components. So the main one and the most important one is basically the chip. And the RIC chip is a silicon microphotonic chip, and it's basically super, super small. In the chip, there is a channel that is carved through it, and it allows the controlled passage of fluids. The setup is also accompanied by a pump and a laser. The pump is a thing that pushes the fluid into the chip, and the laser is the actual thing that detects the passage of each cell. Finally, you also have a RIC analyzer software, which helps with the recording and analyzing the cells afterwards. Now, all this is great, but what is RIC actually recording? So to understand that, we just gotta shift our views. So let's pretend we're looking the chip from the front side. So in such a way that we can see the 3D of the channel that's carved into it. So in the channel of the RIC chip, there are two mirrors. And the two mirrors, actually, there's going to be a laser that is projected. There's a laser light that's projected, and the laser light will start bouncing back and forth between the two mirrors. And this bouncing of back and forth creates an optical phenomenon called resonance. And resonance is great, but it's also just very sensitive to changes in its environment. Specifically, it's very sensitive to changes in refractive index. So just a quick refresher, refractive index is when the light seems to deviate its path when it goes into different materials. It's actually due to a change in speed. One comparison would be simply just a skateboard going from concrete to grass at full speed. Then when it hits the grass, it's actually going to slightly deviate due to its first wheel contacting the grass. But yeah, that's just a comparison for refractive index. The same thing happens with light. And so the idea is when a fluid is pumped through the channel, individual cells pass through, and when the individual cell passes right here, it will change the refractive index because there's a change in material, and this change in refractive index affects resonance. And when resonance is disturbed, well, the whole disturbance is recorded as a parabola, and it looks like this. Afterwards, when the cell passes through, well, it returns to normal. So that's what RIC is basically recording, and this is why RIC is actually called RIC, because R stands for refractive, I stands for index, C for cytometry, and actually cyto means cells, and metry just means measurements. So that's why it's called RIC. Now, of course, there's a lot of cells that pass through that RIC chip at a moment. So we're first to actually plot this into a scatter plot, and each data point will have its associated curve that has been detected. Now, what was the goal of my experiment? Well, there were two goals. The first one was to perform a cell discrimination, basically to differentiate and distinguish different cells from each other, and then calculate cell viability. Now, how did we achieve that purpose? Well, I did an experiment. So we went to the hospital, Maisonneuve Hospital, and we took spleen cells from a mouse. We pumped it through the RIC chip, and we recorded the data. And afterwards, we did a bunch of data analysis. And this is the focus of my project. So I'll break it down into 4 parts for the data analysis. The first part is what I call a parameter tuning. And it's where we take a part of the data that we collected. So we take a sample of it, and then on the sample we perform tuning, parameter tuning. And if the tuning is satisfactory, then we apply the same thing onto the remaining data. So that's step 1. Step 2 is to basically do a full cleaning process. In this case, we have to dig deep into the data and try to find doublets and artifacts, and we have to eliminate them. Step 3 is among the remaining data, we separate the dead cells, viable cells, and debris, so that in step 4, we can take only the dead cells and viable cells to calculate cell viability. Alright, so let's go over the first step called parameter tuning. Now in this experiment, there were actually 4 parameters that were tuned. However, I'll just go over 2 of them called curve start cutoff and curve end cutoff. So what are they? They are basically ways to pinpoint where exactly the parabola starts and where it ends using these 2 vertical bars. So the first one would be the curve start cutoff and this one would be the curve end cutoff. And the purpose is really to isolate where the cell is being detected, because that's what we're interested in. Now this all looks nice, but how does tuning work? I'll just give you an example. So let's pretend this is the data that I collected. To tune parameters, I would start by choosing a point in, so let's say here, and I will open its associated graph, and immediately I can notice that the vertical bars are way off to the side and they're not really isolating the parabola as we want to. So what I would do is adjust the values of each parameter and move them inwards to isolate the parabola. And by doing so, we would eliminate all this irrelevant part. And so this would look like a well-tuned parameter. And yeah, that's it. And so after parameter tuning, we have this scatter plot that remains. So what do we do with that? The next thing would be the full cleaning process. So in this case, we really have to dig into the data and we want to look for doublets and artifacts. Now, what are these? Doublets, they are basically 2 cells that are tightly bound together, and they're usually eliminated based on their larger widths, and they're not relevant to the experiment. That's why we can eliminate them. And finally, we also want to spot and eliminate artifacts, which are computational errors done by the software. How do we do that? Well, for that, we will need more tools. So I'm going to name 3 tools right here. The first tool for finding them is the scatter plot. The scatter plot is really important because it has the ability to switch its x and y axis into about 60 different combinations. And the idea is that by creating all these different combinations, there will be different groupings of cells that are generated on the scatter plot. And by comparing different groupings under different y and x combinations, it's possible then to narrow down similarities between data points and justify why some can be eliminated while others can be conserved. And this idea of grouping is actually so important that we have another tool called viewing options. And in that case, I used the tool of binning. And binning, what it does is basically it colors the grouping. If there are a lot of data points regrouped in an area, then it's going to be colored as red. And then if there are not a lot of data points in one area, then they're colored as blue. And finally, we have the graphical interface, which is what we used previously. That doesn't change. That's another tool. So seeing the graphs. And now how does this all work? The full cleaning process, well, I'll give an example. So this is the data that I have. And immediately when I opened it, I can see that there are very distant points right here. And they basically have a large maximum compared to the other common data points. And because they only represent 0.1% of the entire data, then they're just extraneous points and we can eliminate them directly. However, there are also other points that are scattered vertically, and that is more interesting. So why? To know that, well, we can select one data point. Let's say in this region, I called it region A, and we look at its graph. Well, immediately, this graph right here, we can see that it looks like it has 2 parabolas. And Two parabolas suggest that it's doublets, and doublets, as I said, it's two cells that are tightly bound. And in comparison, this doublet right here, this would be a normal data point, which is not a doublet, and you can see there's only one parabola. So a figure A here seems to be composed of doublets, and actually if we look at all the other regions that I highlighted, so here, here, and here, we can actually see that they all have the same pattern, so they all seem to have to be doublets and have two parabolas. And this can be confirmed due to their percentage. So Rick indicates that all this right here represents about only 3.8%, which is expected for doublets. Like, they're going to be present, but they're not going to take all the space. So 3.8 looks fine. And so we can eliminate these. So we eliminate the doublets and we eliminate these extraneous points, and we get this graph right here. Now, what now? Well, we just did doublets, so now we can do artifacts. So how do we spot and eliminate artifacts? Well, we change the X and Y combinations, and we try to find a combination that gives us interesting groupings. And right here is an interesting grouping, and we can see that they're really high compared to the common data points right here. Why? Well, again, we can select a point, open its graph, and we can immediately see that at this point, the vertical bar, so the start, is here, but the ending seems to be absent. And actually, one could say that it's here, but we're not too entirely sure, so we can manually confirm and verify. So how do we do that? Well, we go onto the scatter plot, and we can see that Rick has associated the full width of this point at about 86. Just for comparison, the common data points in my sample, they're about 50. So really, they're high up there. Why? Well, if we compute manually, so the full width is 86 right here, and then here would be the starting, so 24. And we isolate, so 86 plus 24, we get 110. So this just shows that Rick has calculated the ending of the parabola as 110, and it turns out that it's really just right there. So I highlighted it as a purple vertical bar. And this is clearly incorrect. The ending of the parabola should be like here with the green illustration. And so this is a computational error, which is an artifact, and artifacts must be eliminated. Because really, again, it's not isolating well the parabola. And of course, after that, after eliminating all that, you get this graph right here. And this graph is a lot more interesting, because if my cleaning process was done properly, technically this should consist of remaining cells that are either debris, dead cells, or viable cells. And now one of the last steps would be to discriminate between them, so to distinguish where the dead cells are, where the viable cells are, and where are the debris, so that eventually I can only use the viable and the dead cells to calculate the experimental viability. Now, how do we distinguish these? Well, again, we look at groupings, but actually theory indicates, and a lot of other experiments indicate that viable cells, we know that they are larger. They're larger than debris and dead cells. So just by looking at the y-axis at the full width, we can see that this region, I called it region 2, is way larger than region 1. So theory would indicates that this is viable cells. On the other hand, region 1 is way smaller according to the y-axis, and so it's either debris or dead cells. In this case, it's actually dead cells, and I'll explain why. So if we zoom into region 1 right here, we can actually see that most of its length is about at 35. In comparison, the most common cells, they have a length of 50. So there's a difference. And theory says that for a data point to be considered a debris, it would need to be at least twice as small as what's commonly observed in terms of length. So what's commonly observed in terms of length right here, it's about 50. And so the half of 50 would be 25. However, if you look at 25, there seems to be not a lot of data points below 25. And actually, as I said, they're all at 35 in terms of length. So this indicates that region 1 is not small enough to be debris, and so it must be dead cells. So this is a summary of what I found. So I concluded that region 2 are viable cells, and region 1 are dead cells. Finally, knowing where the dead and viable cells are, we can calculate how much they consist of. So in this experiment, I had about 100,000 viable cells and 16,000 dead cells. We do the experimental cell viability calculation, and we get about 87% of the 87%. And interestingly, if we compare it to flow cytometry, which is the golden standard, like a very reliable and improved technology, it gives us 89%. So really, these results are close. Right. So finally, we can conclude that using RIC, cell viability has been successfully calculated because 87% is really close to 89%, which was obtained by flow cytometry. And because these results are very close, we can also conclude that the cells have been successfully differentiated, notably dead cells from viable ones. Now, one question we could ask is to what extent can RIC actually differentiate cells? Could it possibly identify cells? Well, that's a good question, but that's for the future, and for me, that's it. I hope you guys enjoyed. Thank you.